to welcome Dr. Andrew Chappell. Dr. Chappell is currently the Biostatistics Core Director at the LSU Stanley S. Scott Cancer Center. He's an assistant professor in interdisciplinary oncology department, also in orthopedics. He has an appointment, and he also has a gratis appointment in public health with us. He was formerly with us, and he's moved on to a really nice position in the Cancer Center where he can use his skills in clinical trials. He received his training at LSU initially in the math department, and then went on to get his PhD in biostatistics at Rice University, graduating in 2018. He joined our department in biostatistics in the School of Public Health, and then, as I mentioned, is now the core director at biostatistics in the Cancer Center. So I'm just going to introduce him. Just going to welcome him to give his presentation on current trends in clinical trials. So Andrew, thank you very much, and it's all yours.
Thank you, Dr. Mercanti. Okay, so this is a talk that Don asked me to do a while back. It's current trends in clinical trials, and whenever I was kind of thinking about this talk, I thought about how big the area is. I hope by the end of this talk you have a little bit of appreciation for maybe the infinite magnitude of trial options that are out there, and hopefully that inspires some new ideas and thinking. So let's see if I can press this button. Oh, okay. All right, here we go. Yeah, so as Dr. Mercanti mentioned, I'm the director of biostatistics at the LSU Cancer Center. I'm an assistant professor, gratis. I was here fully at the School of Public Health from 2018 to 2022. Before that, I was a MD Anderson biostatistics trainee, and I did my PhD at Rice. My advisor is at the bottom left side of the screen. That's Peter Thal. He is a pioneer in adaptive clinical trial design from the late 80s all the way up till now. He's a character if you ever get a chance to meet him. And a little about me, I'm an avid traveler. That's me and my wife in, I think that's Florence. I'm a fur dad of Beta and Delta, of course, two Greek mathematical symbols. And now I'm a human dad to little James Genius here in the middle, as 3 months ago. So I don't know if he's watching, but if he is, hey James. Okay, so like I said, I want to kind of give a glimpse of this ocean that is clinical trial design and give some specific examples to kind of pique everybody's interest. So the first thing I did was I went to Statistics in Medicine, which is one of 30 statistical journals that we have. And I just searched, clinical trial design. There's over 7,000 papers on statistics in medicine alone. So in one of our many, many journals, there's 7,000 clinical trial designs. So it's a whole area of statistical methodology. And this whole area is, right, how do we improve our processes for medical advancement, right? So if we're going to move things from the mouse model into humans in early phases, how do we adequately advance through? How do we go to large-scale clinical trials and make the right decisions? So how do we improve that process of getting promising drugs to patients that need them? So there's, a lot of general types of clinical trials, and there's, you can almost kind of imagine a Venn diagram. There are phase 1 trials, which are a small number of patients. They're usually based on safety. A lot of times they're dose finding. So they're trying to figure out among the set of doses for some new agent, what's the best dose. Phase 2 are usually a moderate number of patients. They're usually based on comparing to some kind of standard therapy, usually for short-term response. And then phase 3 is a large number of patients. It's typically survival or response. So, and then there's hybrid versions. So there are phase 1-2 trials, there are phase 2-3 trials. I'll talk about a phase 1-2-3 trial. There's lots of sort of different versions of these. Now, all of these types of trials, all of these general phases, they can be randomized control trials. They can be observational studies. They can be subgroup trials, so in biomarker positive or negative. They can be adaptive designs where you make decisions based on data as accumulated. They can be covariate adjustment. They can be more than 2 treatments. They can be dose schedule optimization. So, chemotherapies or radiations, right? They come in, they get it on different days. Not just how much dose you get, but how often should you get those treatments. They also have enrichment designs, which I'll talk about later, which is basically, can we find a set of patients that most benefit from treatment? So all of these things at the bottom can be nested into the clinical trial designs that we're talking about at the top. Okay, so I'm a statistician. So when I think about clinical trials, I think about a bunch of different parameters that we want to specify. So first, and most important to me, I think, is what outcome is being collected. Is it a survival outcome? Or is it toxicity? Are we interested in continuous data like tumor size, tumor shrinkage, or biomarker responses, or levels of inflammation? What is our variable outcome? Is it your utility score? Is it some kind of a beneficial score that I'm going to spend a lot of time talking about today? Or is it categorical? Other parameters, what's the sample size? Could be something we set ahead of time, right? We could say, hey, we're going to enroll 500 patients, or it could be preset. So, or it could be adaptively chosen. So maybe we say we're going to enroll up to 1,000 patients. We could enroll only 500. We could enroll 1,000, depending on what's happening. What statistical test are we using? What methods chosen? Are there adaptive rules in that? And then I think one thing that a lot of people really focus on and is important is what's the desired type 1 error of the test? So what's the probability of incorrectly declaring a treatment benefit? And the power. So what's the probability under a reasonable sort of assumption, like this drug reduces toxicity rates by 10%? What is the probability, if that's true, that we actually see that result from running your experiment? And these numbers here, right, usually you'll probably see 0.05 for type 1 error, 80% for power, but they really vary. I've worked with a small pharmaceutical company, for example, and they wanted their type 1 error to be 15% because they said, well, if we show a treatment benefit, we're going to sell this drug to Pfizer. So I said, okay. But there's sort of lots of decisions in this, like, what are we going to do with this? They're going to sell their drug to Pfizer. Possibly, if you're working for a pharmaceutical company and you're doing research, you might end up sort of spending millions and millions of dollars on that next phase of research that's wasted. Or you may incorrectly sort of recommend treatments to patients that don't work. Or if your power is not good, you may miss out on opportunities to recommend a new beneficial treatment to patients. Okay, so here's a hypothetical experiment. And from the rest of the talk, I'm going to highlight in yellow design choices. So I want to kind of point out sort of the mix and match. To me, they're kind of like a clinical trial design is kind of like, a lot of Legos, right? You piece a lot of things together. So anything that's sort of a switch out, I'm going to have in yellow here. And it all makes a big decision on your type 1 error, your power, the possible success of your trial. So imagine in truth, God or the universe, whatever you believe in, just told you in a dream that premature delivery rates, if you gave pregnant people this drug, would drop from 10% to 5%. So imagine you enrolled 1,000 patients. So I have that in yellow. Could be 500, could be 5,000, could be 2,000, right? These are design choices. And you randomize patients to the drug, could be observational. And you later observe if their birth is premature. So if it was before 36 weeks of gestational age or not. And you decide to use a two-sided, so I have that highlighted separately, Fisher exact test to determine whether rates differ. You could use a one-sided test. You could use a two-sample test of proportions. You could use logistic regression. You could use a lot of different choices for that, right? And then at the end of the study, you either reject the null or you fail to reject the null, that there's no difference in delivery rates, premature delivery rates by drug. The correct decision, if you know the truth is 10 and 5%, is to reject the null. That's the right thing to do. That's what you'd like to do at the end of this experiment, because then you're going to recommend this drug to reduce premature delivery rates. But that doesn't always happen. You could run the experiment, you might not reject the null. And that is sort of leads into power. Now imagine you ran the same experiment in thousands of clones of Earth. So over on the left-hand side of the screen, these 3 Spider-Men live among different of these multiverse. You run that experiment. And in reality, in every universe, every Earth across this multiverse, the premature delivery rates without the drug is 10%, with the drug is 5%. Power So if you say 80% power, in 80% of those universes, you'll reject the null hypothesis, even though it's true in every single one of them. In 80% of those, if your design is powered to 80%, you will reject that null. So there's still a possibility where you won't. So just kind of it's a theoretical construct of power here, right? And it depends on those things from the slide before. How many patients are we enrolling? What are we doing? Are we randomizing? Is it observational? Are we using a two-sided test, a one-sided test? We're using a Fisher exact test. All of these are design choices here, and that has an impact on it. So generally, right, we want to control our type 1 error, and we want to have a high enough power that if we do have something true, like a 5% reduction, halving of that premature delivery rate, we want to be able to see that at the end of the study and go on to actually giving that to patients. Okay, so parameters matter. Our choice of statistical tests, outcomes, sample size, adaptive decision rules, they all have a big impact on the likelihood you'll publish spurious research. So you'll say, hey, this thing works when it really doesn't, or waste money on follow-up experiments. The likelihood you'll miss a groundbreaking treatment. So if your design is not well powered, maybe it's only powered at 50%, saying in 50% of those experiments, you won't say that there was a treatment benefit, even if there is one. Also, how ethically are you treating patients in a trial? Are you exposing them to a drug that's toxic or doesn't work? And that's why there's thousands of clinical trial designs. So a lot of them are versions, like similar versions of previous ones, where we're just trying to improve these characteristics. We're trying to improve the power. We're trying to improve our control of type 1 error. We're trying to minimize patients that need to be in the trials. And then there's also many, many different flavors of clinical trials. So here's an example of an adaptive clinical trial design. Again, I have yellows here. So simple example of an adaptive clinical trial design, what that might look like. Let's say you randomize patients to 2 drugs, A and B. After 200 patients are evaluated for some binary response. So did they respond, yes or no? You perform a two-sided, 2-sample test of proportions comparing the 2 rates. So do the rates of yes response differ between treatment A and treatment B? If the p-value is less than 0.025, you'll stop and you'll say, hey, whichever treatment has the higher rate at that point, that's the better treatment. Trial's over, okay? That's a superiority stopping rule. Otherwise, if the p-value is above 0.9, you'll stop and you'll declare both treatments equal. So there's no need to continue this trial. They're basically equivalent. You might do that if you have a standard of care, like say aspirin or something, and you have a new treatment that maybe makes patients fatigued, but you think it might improve some outcome. So if it doesn't seem to be improving that outcome, but they're also fatigued, why continue the trial at that point? Otherwise, you'll keep going. You'll enroll 200 more patients. So this is called an adaptive group sequential design, right? So, and again, it depends on these highlighted things. Do we randomize? Could be observational. How many patients do we have in that first bit? Are we doing a 2-sided test or a 1-sided test? What kind of test we're doing? Here we're doing a 2-sample test of proportions. We could have used a Fisher test or logistic regression. We could use covariate adjustment, all these things. And these cutoffs here, this 0.025 and this 0.90, these are also design choices. And we select these because we want to make sure that under realistic ways that the trial could go, we maintain good type 1 error, usually below 0.05, and we maintain good power. So we want to control all of those things with these myriad of choices here. Group sequential designs, right? So it doesn't have to be just 200 and 400. I could keep going to 600, to 800, to 1,000, to 1,200, to 1,400. I could go all the way up to 2,000 patients, continuing this process. I either stop, say one treatment's better, stop and say neither's better, or I keep going. I could also do that at different increments, every 100, every 50, however you want to do it. I'll need to adjust, right, those parameters on the other side to make sure the sample size, the power and the sample size is right. But I can totally extend that across the board. And other decisions I can use is I can adaptively change the enrollment criteria. So I could use some kind of modeling procedure to adaptively decide who's going to be enrolled. I could adjust the randomization probability. So maybe if treatment A looks like it's doing better, more patients are going to get treatment A, but I don't stop the trial, and many more. The point is here, sort of the definition of a group sequential design is we have groups of patients that come in and we sequentially adapt our trial design. So in the other example, right, we sequentially either stop or keep going. We could sequentially change our randomization probabilities, our enrollment criteria, however. And these can be used in any trial setting, and they are. Okay, so you know, I really want to kind of stick to this Pfizer COVID-19 trial. So to me, this is the most famous adaptive clinical trial design to date. And so basically what they did whenever they started this trial, so if you'll remember late 2020, I think they started it, I have a tweet up here. They started it, I think in early September. And basically the way that they ran their trial is they randomized patients to either get the vaccine or not get the vaccine. And then they said, go out into the world, tell us if you feel sick. So they all came back. And once they got a certain number of COVID-19 cases, they stopped and they looked at the data and they said, hey, is there enough data to suggest that the vaccine is working better than the non-vaccinated patients? And if there was, they got some kind of p-value, right? And if that p-value was below some threshold, they were going to stop the trial. Otherwise, they were going to keep going. So they were adaptively making decisions. And if they were going to keep going, they had already vaccinated everybody or given them placebos. So keep going means that they're just going to wait until more patients get COVID. Once they get, 150 cases or 250 cases or 1,000 cases, they had those design parameters set. They're going to stop and look at the data and determine, hey, is there a vaccine benefit? And they were actually powered to 90% to see an efficacy rate of 60% or higher. And if you remember the preliminary trial, I think the efficacy rate was somewhere around like 95%. So they were powered to see an effect that was much smaller than they actually observed in their study. And this worked really quickly. I remember, whenever they were running this, I think it's maybe part of who our president was, but people were really skeptical about it being ready quickly. And I saw that they were using adaptive design. And so I was like, oh no, I think if it works, we're going to see it really quickly. And we did. So yeah, group sequential phase 3 trials, these are in cancer or in vaccines. So in that case, right, we were looking at time to acquisition of COVID. So we were following patients, did it take a month? Did it take 2 months? How long did it take? So if they didn't have COVID at a certain point in time, when we looked at the data, we considered them right censored in terms of survival analyses. This is the same kind of approach that's used for cancer data. If we're looking at long-term survival, so how long did a patient live? If they're still alive, there's still some likelihood that they'll die of some reason, in the future, or progression-free survival or recurrence-free survival, all those things. So these phase 3 trials, they're fully randomized, and we need to demonstrate evidence of a comparative success against the standard of care. So whatever the standard is, right, in the COVID-19 case, we didn't really have a standard. Standard was nothing. So it was against a placebo. But you can imagine a cancer trial, maybe cisplatin is the common chemotherapies that you use, and then there's a new chemotherapy and they test it against cisplatin. So we track patients over time and we record when an event happens. And in the Pfizer COVID-19 trial, again, that was the acquisition of COVID-19. And I have a little highlight here. Weirdly, we wanted people to get COVID-19 because the way that we actually stop and look at the data and determine the rates differ is if we see people get COVID-19. So if everybody stayed home the whole time, nobody ever got it, we'd just be waiting until enough people got COVID-19 to stop and look at the data to actually see a treatment benefit, which you can imagine, after people got certain history exposures, it kind of makes it harder to sort of see that quickly. And after a preset number of events, we stop. So preset number of patients get COVID-19, maybe 500, we'll stop and we'll make that comparison. So for their case, they just used a log rank test. So they compared the survival curves, time to COVID-19 acquisition for the two arms. They got a p-value. They looked at that p-value, compared it to some kind of boundary. Maybe it was 0.01, maybe 0.025. That boundary was set to have certain type 1 error and power constraints. And then they said, hey, either the vaccine works or it doesn't. And they also had a futility rule in there too. So they had a point in the trial where they would say, hey, it doesn't work. We're going to kind of stop observing patients. Okay, so those are phase 3 group sequential trials. That's with survival data. We also have phase 1 clinical trials. So phase 1 clinical trials in cancer, basically they want to look between a set of doses, and I've highlighted here some math. So X1 to X capital J. So capital J is the number of doses. X1 could be like 80 milligrams. X2 could be like 120 milligrams. These are just stand-in for that. But J here is a design parameter. How many doses do we want to test? We want to find the set of doses that has toxicity probability closest to some target. We'll call it pi star. That's another design parameter. By some follow-up time, maybe T. So that could be 3 months, 6 months, 1 month, a year, however long we have, right? Those are all design parameters. And usually people pick pi star around 20%, 25%, 30 or 33%. Now, why do we do this in cancer? My advisor, said something about cancer to the effect of, in cancer, we have to decide whether we cut, slash, or burn. So a lot of cancer treatments are in nature toxic. So there's going to be some degree of toxicity to them. And we understand that. So we want to kind of give a dose level, the goal here, that is acceptably toxic. So, and we're not talking about death here. We're talking about, maybe you have sort of myocarditis, or you have to go to the hospital. Maybe you get pleural effusion where you have to get fluid drained from around your heart or your lungs or something like that. We're not talking about deadly events here, but we're talking about things that are really uncomfortable for the patient, for sure. So we want to target which dose among these doses has closest to pi star. And usually we'll enroll 3 patients. We'll evaluate them for toxicity by some time period. And then we'll enroll 3 more patients based on the data that we had before. So some common designs that you'll see are the 3+3 design. I have a point out here. I just have to point out every time because still like 90% of phase 1 trials use a 3+3 design, even though it has really poor operating characteristics. So what do I mean by that? If we know that the 3rd dose is the best dose, it has a poor probability of picking the 3rd dose compared to these other designs that are out there, but it's still used in most of the time. Up and down designs. And then I'm going to spend some time talking about the continual reassessment method, which is a model-based design. So we have a statistical model to estimate these probabilities. So I just want to give you an example of what this might look like. So let's say pi star is 0.33. We really want to target a toxicity probability of 33% in 5 doses. And let's say these are the true toxicity probabilities. So we just know this. We glimpse into the code of the matrix, and we know that the toxicity probabilities are 10%, 15%, 25%, 38%, and 60%. The one that has the closest toxicity probability is dose 4, right? It's 38%. That's 5 away from 33%. 25% is close. It's 8%, but dose 4 here is the best in terms of what we'd want to target. So ideally, we want to pick dose 4, and we can sort of adaptively assign patients to doses based on estimated probabilities. So a Bayesian phase 1 clinical trial, we assign patients to doses as they come in based on our estimated posterior mean toxicity probabilities. Now, these could be just from a standard frequentist model. We could just fit like a linear curve or quadratic or whatever we want to do to our data that we actually have and get estimated probabilities and do that. The nice benefit of a Bayesian approach is we can use prior clinician belief to kind of guide these probabilities. And that's important because these trials are really only like 30 to maybe 45 patients. They're pretty small. And in the first couple of sets of patients, you may see just by chance 1 dose, even though it's not that toxic, 2 patients have a toxicity, something like that. And we want to make sure we guide this trial. So imagine you have already enrolled 15 patients in the trial. Maybe 3 of them have been enrolled at dose 1, 3 at dose 2, 3 at dose 3, and then the remaining 6 have been enrolled at dose 4. Hypothetically, right? And we've seen toxicities on some of those patients, but we estimate these are our toxicity probabilities: 5%, 15%, 30%, 40%, and 60%. Well, based on these estimates, dose 3 has the toxicity probability closest to our target of 33%. So we'd enroll the next 3 patients that come in to dose 3. And then I'll do this repeatedly. So I'll evaluate the next 3 patients. Now we've got 18 patients. I'll refit my statistical model. I'll get new estimates of this. And then I will assign the next cohort to whichever dose has the toxicity probability closest to that target. There's a lot of versions, so many versions of phase 1 trials in terms of dose finding, but I just want to kind of point out a couple that are famous. The BOIN method, this is from Ying Wan at MD Anderson. Basically, you get sort of like a rule-based design, like the 3+3. So it's not as much kind of staring at probabilities and making a decision. It is a rule-based approach that maybe is a little easier for clinicians to understand. We also have late onset toxicities, so things to account for that. So for example, maybe right after you get the drug, you have some sort of immediate response where you feel sick and you go back to the hospital, but it also may take like a month or two before the effects of getting that chemotherapy start to hurt you, potentially. And so there are models to account for that. This is the low F design, monitoring late onset toxicities in phase 1 trial with predicted risk. And then this second one, Chung and Rick Chappelle, not related to me, from Wisconsin. And Chung is from NYU. This is the tight CRM design, so the tight continual reassessment method. And then we also have things like escalation with overdose control, which is basically, we do dose finding, but we try to sort of be conservative on escalating to the highest dose, unless we have a lot of data to suggest that next step is worth it. Now, all of these designs here are pretty famous, and there is software to use all of them. Here's a shameless plug for one of my designs. This is a subtype design and the 2S subtype design. So this is subgroup dose finding. So imagine instead of just running a trial in one set of patients, we want to look at dose finding in 4 groups simultaneously. And so the design basically compromises between the idea of, these 4 groups have different relationships between the dose and their toxicity probability. And it also adaptively collapses categories together. If the data shows that, hey, patients with this biomarker don't respond differently to doses than patients in a separate biomarker. And the 2-stage design sort of toggles barring and clustering, and I have software here. So this trial was actually used, or this was actually used in a real trial. We have the number here. It was used in child and adolescents for glioblastoma. So the researcher approached me. So he was from Dana-Farber and Harvard. And basically, their sort of assumption was, they were going to give sort of a new agent to these adolescent patients with glioblastoma based on their body weight, right? So there was some kind of normalized based on their body weight. Let's say it was like 80 milligrams per kilogram, something like that, or 150 milligrams per kilogram. However, they had it set up, but they thought that maybe teenagers would respond differently than young children because of additional hormones from being in their adolescence. So they ran this trial using the subtype design as kind of 2 separate groups. It borrowed strength between their dose response curves, and they ran this trial to completion. So just kind of as an example of what this would look like. In their case, right? So let's say we had 2 groups and we had biomarker positive and biomarker negative. And I've enrolled, 30 patients in the trial, maybe 12 in one arm and 18 in another arm. And I get these estimates, these toxicity probability estimates, 5%, 15%, 32%, 45%, and 60% for group 1, and 9%, 20%, 25%, 35%, and 50% for group 2. Well, in this case, right, I'd want to send the biomarker positive group to dose 3. That's the closest to 33%. I'd want to send the biomarker negative group, if that next patient that walks in is biomarker negative, to dose 4, since that probability is the closest. And then once I observe their data, I'll update these probabilities. So every time that a patient walks in the door, I fit the model, I get these estimates, and that's the dose that they get. And if the trial's over, I'd say dose 3 is the best for biomarker positive patients. Dose 4 is the best for biomarker negative patients. Okay, so there are also phase 1/2 designs. So phase 1/2 designs, I'm much more in favor of these than phase 1 designs. These basically dose find between a set of doses, and they want to find a dose with high efficacy probability. So the highest probability of shrinking tumor, for example, shrinking the tumor, or something like that. and they have adequately safe toxicity rates. So they kind of dose find. You want to find the dose level among a set of doses that has high efficacy probability and relatively safe toxicity probability. And it has a trade-off contour. And Peter Thal, this is a phase 1/2 designs. This is the FTOX design. If you go to MD Anderson's website, they actually have a lot of these software. So the FTOX design is one of those. And Peter Thal is a big pioneer in this, and is cited by a lot of different papers. So how do we do this? He initially set up, whenever he did this in 2004, he had like this kind of contour function. You can imagine like efficacy probability, toxicity probabilities. He set up these contours in terms of like what's equally optimal. But in 2020, we kind of thought about this a little differently, of using utility functions. And this is going to kind of lead into a couple designs that use utility functions. So when you're a patient, right, and you get one of these doses, you could have tumor shrinkage. If we're talking about that for efficacy, you could get a toxic response. You could get tumor shrinkage and not a toxic response. You could not have tumor shrinkage and not have a toxic response, or you could not have tumor shrinkage and have a toxic response. So in terms of getting efficacy or toxicity, you can have one of 4 things happen to you. And so the way that Peter really thinks about this is he has clinicians score these outcomes in terms of what's optimal. So the worst thing that can happen is you get the drug, you don't have a tumor shrinkage, and you feel like crap. You have to go to the hospital. That's the worst thing that can happen. That gets a score of 0. The best thing that can happen is the drug reduces your tumor, shrinks your tumor, and you feel fine. That gets a score of 100. What we really rely on is clinicians to pick between these 2 extra scores, which is, what if nothing happens? You don't have a toxic response and you don't have tumor shrinkage. And what if both things happen? You have a toxic response and shrinkage. Now, it depends what we're talking about here, right? If your toxic response is something more like you have a heart attack, maybe it's better for you not to have either of these events happen. But if your toxic response is like general fatigue, maybe you kind of want to favor these. So it's not necessarily that, the yes is better than the no in terms of response. And that kind of depends on how you want to set up the trial. But we get a score, and then we fit a statistical model to estimate these 4 probabilities. And from that statistical model, we can get a predicted score or a mean score for each drug. And we can assign patients to the score that has the to the drug, the dose that has the highest score. So this is kind of introducing you to the utility-based designs. I'm about to overload you with a couple of different ones. There's many versions of this trial using all kinds of different statistical models, covariate borrowing, subgroups, all of these different things. So here's an example with my friend Harry Potter. So we enroll some patients in the trial, and let's say, at this point in time, we've enrolled maybe 40 patients. And I fit a statistical model. I look at, for each patient, did they have efficacy? Did they have toxicity? What dose were they treated at? That's the data to me. I fit a statistical model. I get the estimated probabilities of those 4 outcomes. And from that, I can get a estimated score for each drug, each dose, right? So in the first example, I have an estimated score of 10, 20, 45, 65, and 12. Well, the highest is dose 4. That's 65. So I would assign the next cohort to dose 4. And then, the trial continues, and now I have a new patient come in, and I fit the data again. And now the scores have changed a little bit. Now the scores are 75, 60, 45, 25, and 12. And I assign patients to dose 1 based on that. So why might that happen? You could imagine that, maybe between the first set and the second set, maybe a bunch of patients had toxicities at doses 4 and 5 between that first and second set of estimates. And so it kind of pushed you back down towards the bottom end. So patients are adaptively coming in, they're getting a dose, we're looking at the data, we're adaptively assigning the next patient, we're getting the data, we're making these decisions throughout the trial as they come in. It's not based on opinion, it's based on statistical models and the actual data that we observe. And in the Pfizer trial, they had a phase 2 part, which was establishing dosing in teens. So in the first big Pfizer trial. Utility-based trials. So in general, right, we take a complex patient outcome. In that case, right, we have efficacy and toxicity. Really 4 things could happen based on those 2 things. And we try to get a score. And then we try to sort of assign patients to treatment that have higher scores. So it uses a statistical model to estimate a score for the different treatments groups. Bayesian versions are nice because we can get what's the probability that two treatments differ in terms of their score. We can also get, a probability. What is the probability they differ by 10% or by a score of 10 differential? What's the probability that one group benefits more from a treatment? And many, many more approaches. So using a Bayesian approach, we can actually get posterior distributions on these utility scores as well. So here's another example of a utility score. And this is kind of thinking of it on the opposite. Here, higher score is worse. So this is a clinical trial design based on total toxicity burden. And so whenever they're treating patients for this drug, I don't know what drug it was, they sort of listed all the things that could happen to them on the left. So they see, okay, one toxicity they could have is pericardial infusion, which is where you get fluid built up around your heart. And it could be non-symptomatic, it could require medical intervention, and it could require surgery. So if you have non-symptomatic PE, so you just feel kind of bad, you get a score of 10 added to your score. If you have need medical intervention, you get a score of 60 added to your score. And if they have to operate on you to drain the fluid around your heart, you get a score to 90 added to your score. Now, at the same time, you could also have pneumonia, which would give you an extra 40 points to your score. You could also have myocardial infarction, which would give you a score of 70 added to your score total. So what's actually done in this, now you can do this from a dose finding approach or multiple treatment testing, is we fit statistical models to each of these outcomes based on their dose level or based on the treatment. And we estimate what's the probability for each dose level that we have a non-symptomatic PE versus a mild medical intervention PE versus a surgical intervention PE versus pneumonia, all of these things. And for each treatment or each dose level in the trial, we can get an estimated total toxicity score. In this case, lower score is better. So we want to assign the next patient that walks into the door to the dose level or treatment, if we're looking at multiple treatments, that has the lowest score. Now I'm kind of swapping between these because all of these approaches can be used in dose finding, testing different treatments, however you want to set it up. Here's another shameless plug for a trial that I worked on. This is my boost, because in clinical trial designs, if you have a cool name, that's just what I've always been told is the way to go. My advisor for years and years told me that he wanted to come up with the design name Beat Dogs, and eventually he did have one called B Dogs. He's very, very happy with that. But during COVID, right, early on, there was a couple treatments that were kind of popping around, hydroxychloroquine, azithromycin. Those were kind of popping around early on. And we wanted to run this trial at LSU to pick between those three. So placebo, HCQ, and HCQ plus azithromycin. This was before the FDA kind of came out and said that hydroxychloroquine was toxic to COVID-19 patients. But what we did was we sort of created a score for the different things that could happen to a patient 14 days after being admitted to the hospital. So they could die, or they could be on mechanical ventilation. We gave that a score of 0. You could see the score under UJ. They could be hospitalized with high flow oxygen. We gave that a score of 35. They could be hospitalized with other supplemental oxygen. We gave that a score of 65. They could be discharged with supplemental oxygen. We gave that a score of 75. They could be hospitalized without supplemental oxygen so they can breathe on their own. We gave that a score of 85. And they could be discharged. They're at home. You don't send them home with any oxygen. We gave that a score of 100. So basically what we did was we set up statistical models for these three treatments. This is an ordinal outcome model. So we use proportional odds, logistic regression. And then from these scores, we got an estimated score for each of the three treatments. And as patients come in, right, we possibly dropped arms. We randomized patients to the three arms. And that was how we set this up. But we set up a scoring system similar to the total toxicity burden and the FTOX design. And basically what we did was, I'd imagine, right, we had kind of a drop the loser rule. So if out of the 3 treatments, we saw one treatment was doing way worse than the others, we would use statistics to drop that treatment using the posterior distribution. So imagine hypothetically, patients came in, they had estimated scores 10, 20, and 45 in the 3 treatments. And we have a probability that it provided at least a 10-point improvement. So like a clinical benefit. These scores are between 0 and 100. So a 10-point improvement, you can imagine, in probability would be pretty meaningful. And we basically computed, right, for each treatment, what's the probability it had a 10-point improvement over the worst performing treatment. So in this case, that was treatment 1. So the Bayesian method gave, 50% comparing treatment 2 to treatment 1 and 99% comparing treatment 3 to treatment 1. And we had some kind of threshold, like just earlier before, we had these different p-values for trying to decide to continue the trial or not. We have some kind of threshold here that's set up on this probability so that we have good type 1 error in power. And if that number is high, let's say hypothetically it was 90%, we would drop the worst performing treatment. So we have that probability on the right-hand side is 99%. So we say, okay, we're done with treatment 1. We're going to only randomize patients to treatment 2 and 3 from here on. And we keep going through the trial. And, at another sort of group sequential look to sort of stop the trial or continue, we get estimated scores of 25 and 30. And the treatment that has 30 is higher, right? But it only has sort of about 30% probability of being at least 10 points higher in terms of optimality than the other one. And then we continue the treatment. Now, this also had futility rules in here, and the design could work for up to 7 treatments. So you could sort of play the loser, drop the loser as you went along through a bunch of different treatments. Other examples of utility functions. So it's basically just, like I said, putting scores to things that can happen to patients. Lots and lots of things can happen to a patient for getting in treatment. We could put a utility function, just what treatment gives you the highest restricted mean survival time. You could get a utility function based on complete response, partial response, stable disease, progression, death. You get utility functions based on estimated probabilities of survival at different times. So maybe if you live 1 year, you get a score of 10. If you live 3 years, you get a score of 50. If you live 5 years, you get a score of 100, something like that, right? And we can use statistical models to estimate utility scores for different treatments. You can also use trade-offs like FTOX. Do you want longer survival if you're sick? So there's some kind of like trade-off here on what can happen. And something that people also do is adjust randomization probability. So if UJ is this treatment utility, I can just normalize, right, by all the utilities. So UJ over the sum of all the UKs, sorry, math talk, but if I sort of divide each utility by all of the other utilities, that gives me a fraction between 0 and 1. And I can adjust the randomization probabilities based on that fraction. Okay, so this kind of leads us into another type of trial. These are called platform trials. So this is a trial that's still ongoing. This is I-SPY 2. It's pretty interesting. So basically, this is a trial design that tests multiple treatments at a time against some standard therapy. And they are repeatedly adding new treatments to the trial, and they're repeatedly taking new treatments out of the trial. And they're trying to find benefit in different subgroups like HER2 positive, different biomarker type groups. These are all breast cancers. So again, we have sort of this paradigm here. We get a new participant, they get randomized to the experimental arm or the control arm. Then we observe their outcome. We update our predicted probability. So there is a model that's put into number 3. And then based on that, we either maybe stop a treatment or we keep going, or we stop a patient. And then we're also going to get new treatments put in. So they have multiple things built into this. They have group sequential rules. They have adaptive probability changing. They're also adding treatments into this pot and removing treatments from this pot. They have another nice sort of structure here. So, patients get screened. They get adaptively randomized to one of a couple experimental arms. And they also have a control arm that's always going on. Because basically the idea is, hey, in this case, this control arm, right, is the standard therapy in breast cancers. And we want to kind of test these experimental arms in different patient subgroups. So they get randomized, and then we observe their outcome, and we possibly drop arms. We may add stuff. So Some graduations from this has been running, I think, since 2012. So some graduations have been neratinib has graduated from the I-SPY trial for HER2 positive, HER2 negative trials. It went on to another larger trial comparing that against standard and showed benefit. Another graduation from this trial is volaparib carboplatin, and that was for HER2 negative patients. So all of these sort of are being run by randomizing patients to the different treatments, observing their biomarkers, and then using statistical models to basically point out what treatment should be graduated to that next arm. Umbrella trials and basket trials are like this, but they don't add new treatments. So this is a platform trial. N of 1 trials is also a big area that's starting to crop up right now. And basically what N of 1 trials do is, you know, if I'm enrolled in an N of 1 trial, let's say aspirin versus vitamin D, something like that. Does this help my fatigue? Is, you know, I'll take aspirin for a week. I'll tell them what my fatigue is, and then I'll wait a couple days or maybe another week, and then I'll take vitamin D for a week, and then I'll tell them what my fatigue is, and then I'll wait a couple days, and then I'll take aspirin again for a week. I'll tell them what my fatigue is. I'll wait a couple days, and I'll take vitamin D, and I'll repeatedly sort of switch between taking one treatment versus another treatment and reporting on that. So it's called an N of 1 trial because, like, I'm effectively getting, you know, 5 to 10 different measurements on my outcome for getting the treatment and not getting the treatment in that case. And you get some benefit here too, so of pooling patients together. So you can get really good power from like 10 patient trials, even comparative to like 140 patient randomized control trial, if you get 10 to 15 measurements on those patients. So if you run this trial over a long period of time. And yeah, that's kind of the standard there. And there's statistical models that are going on. And Mamoon, who's here, he's my PhD student right now. He's working on this. We're going to sort of look at putting in group sequential designs for this. But it gives you better power. It allows individualized treatment decisions. And if you can control sort of the carryover effects here, right? Like if you started taking vitamin D right after you took aspirin, maybe the aspirin is influencing your fatigue later on. So you want to kind of control that. But you can get better sort of treatment decisions. And they're doing this a lot in Australia. Another area that is big in clinical trials are looking at dynamic treatment regimes. So this is a dynamic treatment regime. This is an example. Imagine you have an overweight or obese adult. You have no history of, if you have no history of emotional eating, you get IBT. So IBT is individualized behavioral treatment for 5 weekly sessions. And then if you respond at the end of week 5, like you lose weight, you continue the treatment. If you don't respond at the end of week 5, you also get meal replacements. Versus if you do have a history of emotional eating, they get IBT for 10 weekly sessions. If they respond at the end of week 10, they continue the treatment. If they don't respond at the end of week 10, they switch treatment to ACT, which is acceptance and commitment therapy. So these are dynamic treatment regimes. Your treatment changes based on how you respond to that first one. It's dynamic. Now, how did they figure out that this was an optimal thing to do for these patients? Well, they used a SMART design. So SMART designs are clinical trials built for dynamic treatment regimes. So you could imagine that we get patients in, we randomize them, that says R to IBT for 5 weekly sessions, or we randomize them to IBT for 10 weekly sessions. And then we say, okay, did they respond at week 5? If they respond at week 5 and they had the 5 weekly sessions, we continue their treatment and we observe their weight loss, something like that. If they didn't respond, we give them IBT and meal replacements, or we randomize them, see that big R there, we could randomize them to ACT. And if you're randomized to the 10 weekly sessions, if they responded at week 10, you could continue the treatment. If they didn't respond at week 10, you might randomize them to also getting meal replacements or switching them to ACT. And then in each of these treatment arms, right, we observe their data, what was their end up weight loss, and we can sort of decide for, even in the previous example, different subgroups of patients, which sort of treatment regime operates the best. Enrichment design. So enrichment designs are aimed at finding or sort of enriching patients based on the data that we're collecting. So we might be running a trial and then we might see, oh, patients with this one biomarker, so HER2 negative, benefit from a treatment more than the other patients. So enrichment designs not only are testing whether the treatment works overall, but they're trying to highlight sort of biomarker-driven areas where patients could most benefit from a treatment. It's kind of a personalized medicine approach. This is a relatively new area. They are doing more of it at MD Anderson. Other outcomes you could use. So you could use designs based on longitudinal continuous outcomes, right? You could get repeated measures on BMI and estimate polynomial curves and see if Ozempic, for example, changes that curve relative to placebo. You could look at longitudinal A1C in patients with HIV and see whether different treatments sort of change sort of that longitudinal trend. So you could even look at sort of like estimated curves based on repeated measurements. All of these have to have like a statistical model set up underneath, right? You could look at multiple different types of events. So different types of toxicity, a headache versus a coma. Maybe you want to control for both. Maybe you want tumor reduction and overall prognostic benefit. You can also look at multiple transition times, right? So in cancer, a patient could progress and then their tumor could shrink and then they could go into remission and then it could come back and then they could die, right? So you can have sort of all of these different transitions that could happen as far as a clinical trial design. And you could set up trials sort of based on, maximizing the probability that you go into remission and stay in remission, or minimizing the probability that if it comes back, you die quickly after that. And you could set up times based on this. It's kind of infinite in a way, right? And then lastly, I just kind of want to highlight a new area of thinking, which is kind of turning the whole thing on its head. So right now, the conventional way that people do cancer trials is they do a phase 1 trial where we have some agent A, call it agent A, it's our experimental agent, and B is maybe the control, like what's the historical control. And we are using a phase 1 trial to pick a dose level. So it could be dose level X2 up to XJ. We pick a dose level to go on to new trials. So we go on to a new phase 3 trial, and then we do a survival comparison. Okay, survival comparison of what we picked in phase 1 or phase 1-2, however we set it up, versus the standard therapy. And we want to see basically that new treatment provides, let's say hypothetically, a 12-month improvement in survival. Okay, and we'll do a hypothesis test and we'll see, does it actually provide improvement? That's the standard therapy. We pick a dose based on a small number of patients. We go to a large scale trial and we test the thing that we chose for the new agent against the standard. And then we see, does it actually give you a survival benefit versus what's usually done? Now, unfortunately, about 50% of phase 3 cancer trials give a negative result, which is crazy. So, these are multi-million-dollar trials, hundreds of patients. So one way that we're kind of thinking about it is allowing for re-optimization steps. So what if you pick a dose in phase 1 that's suboptimal in terms of survival? Maybe it has decent toxicity probability, but it doesn't actually increase patient lifetimes. We have sort of approached a design called a phase 1-2-3 trial, which will switch doses in that phase 3 portion to a dose that maximizes survival based on the patient data that we've already selected. And then you'll continue the trial. This is a paper, this one, 2019 paper of the year in biometrics. And just to kind of illustrate it, imagine in phase 1-2, we have 5 doses. We pick dose 4. So we start dose 4 versus the control, long-term survival. We start running the experiment, and then we see that dose 3 actually looks like it has the best survival time compared to the other doses. Well, what this design will do, and that's based on the phase 1-2 patients, this design will switch doses from dose 4 to dose 3, and then it will finish the group sequential design comparing dose 3 to the control. Also, another example, let's say we pick dose 3 and we start randomizing patients between dose 3 at the new agent and the standard, and it looks like actually dose 3 still has the best mean survival time. Well, then we just finish the trial the way we would normally do. And we showed that this design, this sort of re-optimization scheme, actually improves the probability of like declaring a treatment benefit by anywhere from 20 to 80%. It's pretty substantial across different scenarios. So there's a drive right now thinking about sort of, instead of segmenting these into phase 1, phase 2, and phase 3 trials, sort of thinking about a comprehensive approach to, in the end, what we want is we want to get a drug, a new cancer drug that improves patient lifetimes. That's what we want. So thinking about this all together, there is a push for that now. The future, it's bright. There are thousands of clinical trial designs that could be used for real trial. Most of them have user-friendly software and a methodology paper. So you can talk with your statistician about implementing these type of things. And if you meet with a statistician and you explain your idea carefully, they could come up with a new trial just for what you want, a new methodology to tackle that. And I also have a line here that somebody has mentioned to me, which is algorithmic decision-making. It might also help with patient enrollment, sort of due to increased trust, right? So they're going to get at any point in time in the trial, they're going to get the treatment or the dose level that gives them the best probability of having a good outcome in their disease. Yeah, data-driven decision-making in the future. So we have self-driving cars that uses data to make decisions on how to drive. Oil is using data to drill and try to identify pockets of oil. If you use Google, you use the Google bar, it pops up a whole bunch of search options. So all of these things are using data to adaptively make decisions. Why shouldn't medicine? And last plug that I always like to have in my talks, make sure you work with a statistician. Statisticians can help you think about a lot of different things that maybe you haven't thought about for your study. They can also use new statistical methods. And a famous quote, asking about a statistician for help after the data is collected, he can perhaps say what the experiment died of. And that's it.